Hi guys, in this tutorial I'll show you how to browse GeneProf datasets, how to export data and how to generate plots. As an example, we'll use some data from an experiment by Rada Iglesias and his colleagues, in which the guys have studied gene expression and transcription factor binding in human embryonic stem cells and neuroectodermal spheres. On the experiment main page, I'll be looking for the main results section, and I'll specifically deal with this dataset that has the combined ChIP-seq and RNA-seq data in it. It's a blue dataset, which means it's gene-centric data, and I'll first open that in a new page. Now we can see this dataset contains a whole lot of different columns. There are gene expression values in reads per kilobase million format, logarithmic fold changes, p-values, and a number of little texts that represent binary associations between ChIP-seq peaks and their target genes. Let's start by plotting this data. Now at the bottom of this page there's a little button that says plot data. When we click that, we get a menu with different plotting options. We can plot scatter plots, histograms, Venn diagrams, box plots, and a number of other things, heat maps. Let's just start with a little scatter plot. Now we have to select the data that we want to include in the scatter plot. GeneProf has made a little default selection there, but we only want to include the gene expression values in stem cells and in neuroectodermal spheres. In this case, there are a number of options to customize the plot, but all we want to change is to use a logarithmic scale, which generally works out looking better for gene expression values. Click plot, and a few seconds later GeneProf should have prepared the plot for us. There it is. So you can nicely see the scatter between embryonic stem cells and NACs on the y-axis, centered around an equal diagonal represented by the red line. So it gives us an idea about how similar the gene expression is. You can also plot Venn diagram here. Let's pick as an example the binding of the transcriptional co-activated P300 in ESCs and in NECs, and see how well they correspond in the two different cell types. So I just enter labels and select the corresponding columns and click plot. Leave all the other default options, and again a few seconds later GeneProf should have generated a plot for us. Yeah, takes another moment. There we go. So we see P300 binds to a whole lot of genes in embryonic stem cells, to slightly fewer genes in NECs, and there's a considerable overlap. However, there are 852 genes which are bound by P300 in NECs, but not in H9 embryonic stem cells. So let's find out which these genes are. So I'll first close all the plotting dialog, and I'll open a dynamic table browser. Now this displays all the data in a paged view, and we can change which columns are displayed by clicking the display options. And I just first hide all the data I'm not interested here, like the p codes for various histone marks, TFA scores, which we're not working with now in this example. And instead I'll add some additional columns, general annotation columns that are available for every data set. So I'll add a feature type, sample gene ID, and description. Reorder that in a nice way, and then click apply. And then we can see a new table here. It's a bit more concise. And we can now proceed to sorting this by just clicking a column header, sorting here by the logarithmic fold change, or by the name, or we can filter it. So let's filter for genes that are bound by P300 in NECs, setting this filter to true, and that are at the same time not bound by P300 in H9 cells. You can see at the bottom left of the page there's a number 852. That's the 852 genes we saw in the Venn diagram earlier. Let's find out which of these are also differentially expressed. So I first filter for those genes that have a p-value of smaller than 0.05 and also a logarithmic fold change greater than 1, so about a two-fold change, that is. It's like the absolute fold change, so that means either up or down regulated. That's the result, there are only two genes. Right. So finally, let's look at how we can export this data in case we prefer to work with it in, say, Excel or another external application. So we can export every GeneProf dataset in text file format, as a spreadsheet, in XML format, or in an R data format. You can open directly in the R software. Yeah, so we can just export a spreadsheet, open that, and there we have exactly the same data we were looking at just now in Excel. So you can work with it there if that's what you prefer. For other data sets, like genomic data or nucleotide sequence data, there are other data formats available in addition to these that might be more appropriate. For example, this is a genomic data set. We can export the data in BED format or in big format. And I'll show you an example of a sequence dataset as well. We have additional options available here, the faster or FastQ format files. Alright, this is the end of this tutorial. I hope you guys have found this helpful, and see you next time.